Hi everyone. Today I am going to discuss about the structure of nucleus. Okay. So as already we know about the cell. The cells are the smallest functional unit of living organism, and they have three main components regardless of their types. Okay. Number one is in plasma membrane. Number 2 is in cytoplasm. Number 3 is nucleus. Whatever type of cells they are, this cell will have these three main components. Okay. So this is one of the cell organelles of the cell. What is cell organelle? Okay. Cell organelle means little organ. The cell contains many little organs, okay, which are involved in particular functions. So each cell organelles involved in specific functions they have to perform. Okay, so thereby that cell contains many cell organelles which are part of the cell and do their specific function. Okay, so nucleus is considered one of the important cell organelles. Why? Because it is considered the brain of the cell. Why do we call it the brain of the cell? The nucleus control all the cell activity. Okay, the nucleus control all the cell activity. So all the cell activity is decided by the nucleus. Why? How it will be decided by the nucleus? Because this nucleus is having a nucleolus, okay, and as well as the chromatin fibers. That is a genetic material. Okay, so this genetic material decide what the cell need, what the cell has to do, or what type of function it has to perform. All these things are decided by the nucleus. So that's why we call it a nucleus is a brain of the cell. Okay, let's enter into the structure of the nucleus. So the nucleus is made up of double membrane structure called nuclear membrane. Okay, the space between the two membrane is known as peripheral nuclear membrane. Peripheral nuclear membrane. So this nucleus is surrounded by the membrane, double membrane called nuclear membrane. The space between the two membrane is known as a perinuclear membrane or a perinuclear space. Perinuclear space. Okay. So followed by that, we will identify on the surface of the nucleus numerous pores are identified. These numerous pores are nothing but the nuclear pores. The nuclear pore is surrounded by the structure called annulus. So singular form is annulus. Okay, so nuclear pore is surrounded by a structure called annulus. So through this pore, mRNA, protein, lipids, macromolecule substances are able to enter inside of the nucleus and able to come outside of the nucleus. So entry and exits of these macromolecules, protein, enzymes, mRNA are done through the nuclear pore. They enter into the nuclear pore. They exit through the nuclear pore. So nuclear pore acts as a gate for the nucleus. Okay, through that only that all the mRNA, protein molecules, macromolecules, enzymes, all these substances are able to enter and exit of the nucleus. Okay, so next things we are able to see the nucleoplasm. This nucleoplasm is a gelatinous matrix which contains the network of the chromatin and a small spherical shaped structure called the nucleolus. So this nucleoplasm contains a gelatinous matrix where this chromatin network as well as nucleolus are situated. Okay, so look at this small ball like structure, that is we call it a nucleolus. It may be many or numerous nucleolus or one nucleus. Numerous nucleolus are one nucleus. Okay, the nucleolus may be many or single. So single form is termed as a nucleolus, and many forms is termed as nucleoli. Okay, so thereby this is the cell may have one nucleolus or many nucleolus. Okay, many nucleolus is termed as a nucleoli. Clear? So what is the function of this nucleolus? This nucleolus is the site of the ribosome synthesis. So thereby the nucleolus doing biosynthesis of ribosome. So what is the function of this ribosome? This ribosome is the site of the protein synthesis. Ribosome bind with the mRNA and exit the nucleus, enter into the cytoplasm where this transcription is done. Transcription as well as the translation is done. So this mRNA bind with the ribosomes. So like that. This ribosome is having the two units. One is a small subunit and large subunit. Okay. So this mRNA bind with the ribosome, and then it involves the transcription process as well as translation process. Okay, mRNA code for the protein, then this protein is synthesized by the translation process. That we will discuss in later on. Okay, because for the preparation of the proteins there are three steps: DNA replication and mRNA, that is the transcription, and third process is translation. Replication, transcription, translation. Translation is the final stage where the protein is synthesized from the coding of the genes. Clear? So we will discuss that one later. So the nucleolus involved in the ribosome synthesis, which bind with the mRNA, involved for the transcription process in order to produce proteins through the process called translation. So thereby this ribosomes is the site of the protein synthesis. Okay. So when these ribosomes are synthesized in the nucleolus, they enter through the folded structure called, that is an endoplasmic reticulum. This endoplasmic are bounded to the membrane, that is a nuclear membrane. So through this endoplasmic reticulum, these ribosomes are released. Okay, so some of the ribosome stays on the surface of the endoplasmic reticulum. So thereby it is known as RER. If the endoplasmic reticulum possesses ribosomes on its surface, then it is known as RER, nothing but rough endoplasmic reticulum. If the endoplasm is not possessing the ribosome, then it is known as SCR. So RER involved in the protein synthesis and SCR is involved in the detoxification of the toxic substances and membrane biogenesis process. So SCR involved in the membrane biogenesis process and detoxification of the toxic substances. Okay, so let's discuss about the chromatin. This chromatin is uncoiled, indistinct in the nucleus, okay, which is later on condensed into the chromosome. During the interphase of the cell cycle, it is look like that network structure called the chromatin. Okay, so once this chromatin enter in the prophase in the cell division or cell cycle stuck, then it is condensed to form the chromosomes. Okay, till that it will be uncoiled, indistinct thread-like structures. Okay, so once it enters into the cell's division, chromatin condenses to form the chromosomes. Okay, this chromatin contains little amount of the DNA and RNA and histone and non-histone proteins. Okay, so with this chromatin network, the DNA coiled around the 8 histone proteins. 8 histone proteins. So, okay, so making the coiled structures. So if you take and if you keep 8 balls and on the surface of the 8 balls, if you coil the thread, how it will look like? The same way these histones are coiled by the DNA structure, which is the repeated units. Okay, so repeated units, that is the DNA structures, and it forms the repeated units, that is called a nucleosome. The unit where that 8 histones are coiled by the DNA structure is termed as a nucleosome. Okay, so along with this, the chromatin along with the histone proteins, they are also having the non-histone and little amount of the RNA and DNA. Okay, let's move on to the structure of chromosome. Okay, the structure of the chromosome. Before that we will see what is a euchromatin, what is heterochromatin. Okay, so the chromatin condensed to form the chromosome structure. Okay, this is a centromere. This one is an chromatid. Okay, that's an chromatids. These are sister chromatid. Got it? So here you will find it that is, we call it an pentochore protein. So where this spindle fibers are attached, clear? So here this chromatid is having the two portions. One is an heterochromat. Okay. So this region is heterochromatin. Heterochromatin does not have the gene for the mRNA transcriptions. So heterochromatin does not have the gene for mRNA transcription. So thereby they are tightly packed. They are tightly condensed. Okay. So and in case another region, that portion we call it an euchromatin. So this euchromatin has the gene for mRNA transcription, so thereby it is loosely condensed. It is loosely condensed. Okay. Then the tail region, the tip of the chromatid is known as telomere. This is known as telomere. Okay. So the importance of telomeres are identified in recent times because this telomere is having the tag sequence. Okay, the telomere is having the tag sequence and which is responsible for the aging of chromosome. So it is involved. Then the recent studies reveals that the telomeres involved in the aging of the chromosomes. Okay, this telomere it prevents the fusions with the other chromosome. So this telomere prevent the fusion with the other chromosomes. So that's the major importance of the telomere. One is that it is involved in the aging process of the chromosome. Lifespan of the chromosome is determined by the telomere. Lifespan of the chromosome is determined by the telomere. So lifespan of the chromosome is determined by the telomere. So it is involved in the studies of aging of the cell as well as the cancer. Okay, aging of the cell as well as the cancer. It is having the sequence that is an tag sequence. Okay, so it prevents the fusions with the another chromosomes. Okay, this is the chromatin structures we have seen, and in the next video we will discuss about the chromosome and its structures and the position of the centromere. Thank you for watching my channel.